Hello everyone, and welcome to Sabrina Teaches. Our topic for today is oncogenes, but before we begin, we need a foundation. Our body contains trillions of cells that work together to keep us healthy and functioning. Now, what is a cell? Cells can be viewed as building blocks that provide structure and function for living organisms. Every cell in the human body contains 46 chromosomes. These chromosomes carry DNA, deoxyribonucleic acid, and a small section of the DNA is the gene, which comprises small chemical bases called nucleotides. These bases are A, C, T, and G, and they form the DNA double helix structure. Each human being contains approximately 20,000 genes and 3 billion nucleotide bases, which makes your genome. Proto-oncogenes are genes responsible for regulatory proteins, which help regulate the region of the DNA that's copied. They're also responsible for growth factors and growth factor receptors. Growth factors will bind to the receptors on a cell surface and initiate a cascade response resulting in growth. Additionally, proto-oncogenes code for proteins responsible for cell division, cell differentiation, which is when cells mature to perform their specific functions, and cell death, also known as apoptosis. The functioning of proto-oncogenes is necessary for human development and the health of tissues and organs. For instance, embryogenesis being the formation of an embryo. However, a single mutation or change in the nucleotide sequence can cause previously normal cells to now become cancerous, and this mutated version is known as an oncogene. If this single mutation is not repaired, it will be passed on to daughter cells. These oncogenes will remain on and always functioning, leading to more cell division, less cell differentiation, and inhibiting cell death, which may cause abnormal growth or cancer. An analogy that can be made to understand this better is a train running in the TTC, Toronto Transit Commission. Imagine the TTC as the genome, the train as a single strand of DNA, and the train operator as a nucleotide base. Replacing the TTC operator with someone inexperienced can harm the functioning of the TTC trains, including speeding, missing stops, and crashes, just as a point mutation on a DNA strand can cause the formation of an oncogene, which contributes to the development of cancer. Oncogenes can be activated in several ways, including epigenetic changes, chromosome rearrangement, and gene amplification. Epigenetic modifications include the addition of a chemical group being added to the genetic material, so the DNA, which can cause oncogenes to be turned on. Chromosome rearrangement can occur during cell division when DNA pieces from one chromosome are transferred to another. This change in DNA sequence can lead to the activation of an oncogene. Lastly, gene amplification is when the genome of a cell contains excessive copies of a gene, leading to too many protein products. Ultimately, excessive protein production, decreased cell differentiation, increased cell division, and the suppression of apoptosis are defining characteristics of cancer.